Yeah, ready.
Thank you. Hello, welcome to Become a Citizen Scientist in New South Wales. I'd like to begin by acknowledging the traditional custodians of the lands we're meeting on today. I personally am on Darkinjung country, and I pay my respects to elders past and present, to the Darkinjung people, and also First Nations Australians all the way around the country, and indeed Aboriginal persons all over the world, and acknowledge that sovereignty was never ceded. Um, we are all here today to learn a bit more about citizen science, specifically in New South Wales. Um, my name is Lynette Plenderleith. I am a committee member of the Australian Citizen Science Association. I'm an ecologist and a teacher and all kinds of science communication-y type things with a bit of a background in citizen science myself. But I'm excited to hear about some specifics about New South Wales citizen science. I've only been living here the last 18 months. So there's lots of interesting things that are going on that we are all looking forward to hearing about tonight. Just a reminder, this is being recorded. If you don't wish to be recorded, then you're welcome to change your name so that you don't have to appear as your name that you're currently in our system, or you're also welcome to turn your camera off. Whether you have your camera on or not, please remain muted unless you're invited to speak, because otherwise we get a bit of interference and it makes it quite distracting for all involved. Um, we also though do encourage you to ask questions, but not by shouting out, but by typing them in the chat. So Larissa and Thomas, I believe you're both going to be able to see those questions and happy to answer them as we go along. And then at the end of each presentation, and then there should be time at the end for us to have a bit more of an informal discussion where you can choose to put your cameras on, your microphones on, and have a bit of a chat. Answer some more questions, have a bit more of a chat about citizen science in general and in New South Wales. So we are going to have a talk by our esteemed committee member and president Larissa, who is going to talk about citizen science in general and in some specifics about New South Wales citizen science. And then we have Thomas, who is also on our committee, who will be talking about iNaturalist specifically and contributing to citizen science in your own time from home as a obsession for me personally, but as a fun and relaxing time and way to contribute to science through the citizen science platforms. All right. So we also have Alice Howe, I believe, in our audience today. So Alice Howe is our Secretary of Australian Citizen Science Association New South Wales chapter. We have Larissa Braz-Souza, who I'll be introducing formally more in a moment, and Thomas Massalio, who is also on our committee. The committee's been formed since May 2024. So the Australian Citizen Science Association New South Wales chapter is quite new still. And we still are very much welcoming of feedback and ideas, suggestions about things that you might like to see and do around citizen science in New South Wales. So I will introduce our first speaker, Dr. Larissa Braz-Souza, a biologist, researcher, and associate lecturer at La Trobe University. So Larissa was a New South Wales resident until fairly recently, when she popped down to Victoria to take a job at La Trobe University. But her PhD is on mosquitoes and public health, and she leads the Learning by Doing team at the University of Sydney, working with schools and communities to make science hands-on, local, and meaningful. She's also co-chair of AXA New South Wales. Larissa co-founded the Great Southern BioBlitz and coordinated the National Mozzie Monitors Programme, empowering people of all ages to explore biodiversity and contribute to real research, from backyard insects to large-scale community bioblitzes. More about bioblitzes and what that actually means in a moment, I'm sure. Her work brings creativity, curiosity, and connection to every project, inspiring thousands to discover the science in their own local areas. Larissa is a personal friend of mine. I am constantly and consistently impressed by her work. I admire her as both a person and a scientist, and I'm very excited to hear what she's got to say about becoming a citizen scientist in New South Wales. So Larissa, when you're ready, thank you very much.
Thank you. I got so happy with this introduction. Thank you. So welcome everyone, and thanks for joining us for this webinar today. This webinar is presented by AXA New South Wales, and as Lynette was saying, we are a very new establishing chapter. So the Australian Citizen Science Association has been running for 11 years now, and we are an association that brings together different citizen science projects, project leaders, citizen scientists, researchers, educators, people interested in citizen science, to have a place to talk about it, to increase the visibility of projects, to talk about the national strategies to bring citizen science to people's everyday lives. So it is an honor to be part of the establishing chapter in New South Wales. I was living in New South Wales until 4 months ago, 5 months ago. And but before that, I lived in South Australia for 4 years. Yeah, I've been in Australia for 7 years, so I've been moving a lot. But I had the chance to be part of the local chapter in South Australia, and I learned a lot as part of the local chapter, and it was a great opportunity to connect with local projects. So I'm very happy to be part of this establishing chapter now and help connect people with local projects. So today, whoopsie, go through it. I have just a few slides. The idea is not to run a lecture about citizen science. They are like 10 slides maximum. I have a lot of photos of projects, but I'd like to share some insights into what citizen science is. I know some of you already have experience with the citizen science. Some of you might be joining or wanting to join a citizen science project for the first time. So I have some examples of what it is, some benefits and discoveries from around the world, but mainly from New South Wales. So, according to the Australian Citizen Science Association, citizen science is the public participation and collaboration in scientific research, aiming to increase scientific knowledge. I really like to bring this definition every time I talk about citizen science because it's much more than outreach, for example. It's much more than participating in a project for, like a few weeks, but then you don't know exactly how you are contributing. So in a citizen science approach, not we have, but like we usually increase scientific knowledge. We work on making scientific discoveries together. And that means that people don't have to necessarily be a professional scientist to become a citizen scientist. Um, so that's an opportunity for people to get connected with researchers, other members of the community who are also interested in similar projects, and to learn something new, but also to discover new things. It could be discovering new species. It could be, yeah, there we go. We have some examples here, discovering new planets. And that's been happening for at least 90 years with the citizen scientists joining citizen science projects organized by NASA, discovering new molecules or helping make new drugs. For example, as part of Breaking Good, which is a chemistry citizen science project. It's organized by the University of Sydney and it runs in New South Wales. And every year we have year 12 students coming to the university labs making new drugs or drug candidates that could treat neglected diseases like malaria or mycetoma. So that's an example of, chemistry citizen science project. So citizen science is really, really diverse. So most projects are around biodiversity, are focusing on biodiversity monitoring and management. But there are citizen science projects in lots of different areas. So chemistry related, or molecular biology, or astronomy. Fold It is a very interesting project. I always, like to talk about it just quickly here, because it is an uncommon project comparing to the other projects about monitoring, for example. With Fold It, people can play games like video games, but these games they are actually simulations of how proteins could fold inside of our bodies. And depending on the temperature, depending on the exposure to other proteins or antibodies or drugs. And then Folded has been running for years as well. It is a US-based project. It is global. You can play from anywhere if you're interested, but it started in the US. And the chemist, the researcher responsible for designing folded, won the Nobel Prize last year for his work with folding proteins and protein design. So it is an amazing project, and people also played this project to help develop new possibilities during the development of the COVID vaccine. So we can see like there are a lot of applications for citizen science and many different ways to get involved. So yes, around the world, people are discovering new drugs, diseases, molecules, species. We have some examples from species in Australia, uh, planets, comets, stars, galaxies, from getting involved in citizen science. And there are, again, many different ways to get involved. Every project will provide a different opportunity by counting species, by playing games, for example, by being out in the field, taking photos and reporting using a specific app, collecting water, samples of water. In Australia, we have Water Watch, for example, and reporting those observations to scientists. So there are lots of benefits for scientists, but also for citizen scientists to get involved. In Australia, yeah, we have a breaking good, we have Mozzie monitors, Great Sound and BioBlitz, Frog ID, which was designed, developed and run by Australian Museum, one of the largest citizen science projects in Australia, Invertebrate Australia, which organizes a lot of different citizen science projects, including the Christmas beetles. Christmas beetles count that is happening right now, and you can contribute observations to iNaturalist. So I wanted to quickly talk about the two big citizen science projects in Australia. One of them is the Mozzie monitors. So we have like a short article here. And another one is the Aussie Bird Count, which this one is the largest citizen science project in Australia. And this project is seasonal, so always runs in October. And this year we had more than 5 million birds counted. And people can observe birds from their backyards. It doesn't have to be from the backyards, but you can observe birds from anywhere for 20 minutes. You count the species. The app is very interactive, so if you're not sure of the name of the species, you can use the app to find out. So you can filter species by the shape, by size, by color, and then you can identify the actual species you are observing, and then you can enter the species on your count. And it's very valuable, the information we collect from here, because over time we can see how many birds people are observing in urban areas or in national parks, for example. And we can see how much people are, how much interaction people have with those species of birds. If the observations are increasing or declining. So that's all valuable information that we have when people participate over time in that kind of citizen science project. We usually have, like threatened species, rare species of birds counted, and then it's again valuable information for researchers to know where those species are when people are sharing. And Mozzie monitors, which was, we had the logo in the other slide. That was actually part of my PhD. So it's still running, sorry. That is a citizen science project where we provide mosquito traps for people interested to set up these traps in their backyards, collect mosquitoes. They use a specific tip card that we provide. They take photos of mosquitoes, and then they share the photos with us, with researchers. So we can count, we identify the species, and we let people know what kind of mosquitoes they have in their backyards. So this project has been running since 2018. So it's been 7 years now. And we had a lot of discoveries with Mozzie monitors, but something that was very interesting for me in the first year, as part of my PhD actually, I compared data collected by Mozzie monitors with the data collected by a traditional, professional mosquito surveillance project in South Australia at the time. And for the same period between October and March, which is the peak season of mosquitoes in Australia, Mozzie monitors collected 4 times more mosquitoes than the professional mosquito surveillance project. And it was almost the same diversity of species, but in terms of abundance, Mozzie monitors collected 10,000 mosquitoes in 6 months. And then the professional mosquito surveillance project collected about 2,000 mosquitoes, over the same period. And I think it is a great example to show the power of citizen science by increasing, upscaling our ability to collect data from places where, scientists can't be, or, technicians can't be, at the same time. And there's another way to participate in Mozzie monitors, which is also by using iNaturalist. Thomas is going to talk everything about iNaturalist in a couple of minutes. And so from using iNaturalist, people can take photos of mosquitoes from anywhere in Australia at any moment and share their observations on the app, upload the photos on the app. And then in early 2021, during Mozzie Month is between February and March, which is the end of the peak season for mosquitoes. We had the upload of one specific species on my naturalist that was in far north Queensland, that was in Cape York, in a national park. Uh, and that was by a citizen scientist using a naturalist. And then we discovered that was the first detection on the first occurrence of that specific species in Australia. It had only been observed in Papua New Guinea almost 100 years ago. It had never been reported in Australia, but it was reported for the first time by a citizen scientist using a naturalist. And again, I think it's another great example to show the power of people getting engaged with the different citizen science platforms, but also to show the power of people's observations. So every day, things, species, planets, plants, everything we are observing has a value if we share. And then citizen science brings that possibility for people to share with the researchers, with the scientists, with the broader community. So I brought just quickly, a few examples from New South Wales here. And this book was just published, and Thomas is one of the authors. And that's a field guide to fishes, different species of fishes in Sydney Harbor. And something that Thomas was sharing with me a few months ago. More than 10% of all the species recorded in the area, they have only been discovered in the last 10 years from observations on iNaturalist. And again, iNaturalist is a free-to-use citizen science platform. Thomas is going to talk about it, but it is a way to make science more accessible. So if you have a smartphone, you can use the app, you can take photos, and you can share with other people. And you have almost instant feedback for your observations. So the app suggests you some possible identification. So there is an AI, Thomas will talk about it, but there is also verification from other people in the platform. So you might have the opportunity to actually collaborate and talk with researchers if they are coming to validate your identification, or they might also come and talk to you. There is a kind of a chat embedded on iNaturalist. So it's an opportunity to connect and also to share your observations. Uh, this is also from iNaturalist, and this is a rare observation, not observation, that's a rare species, threatened species, a pink slug that's only found in the north coast in New South Wales. And that was observed for the first time after the bushfires between 2019 and 2020. And it was part of the Citizen Science Environmental Recovery Project. So that was observed by a citizen scientist, and he shared his observation on iNaturalist. We're going to share those slides at the end so you can check the observation here if you want. There are other amazing photos from that species. And it's a very hard to spot species, and especially after the bushfire, researchers were unsure of the status of the species after the fires, but then it was observed by a citizen scientist. And again, lots of species that were unseen for 100 years. So here we have species of grasshoppers. And again, lots of observations from iNaturalist, from people uploading observations on iNaturalist, and including photos of living species, specimens for the first time. Some of these species they were only observed, they were only available at museums, like as an entomological collection. So they were not live specimens. But here on iNaturalist, we had first photos of living individuals shared on the platform. And here I know, like I brought all examples from iNaturalist, and we're going to talk a lot about iNaturalist today because it is an accessible way to participate in citizen science from anywhere. As Lynette was saying, it's her favorite way to participate because you can join from home. You can take photos from your backyard. You can record sounds from your backyard. But there are a lot of different citizen science projects going on in Australia. Was talking about frog ID from the Australian Museum. There were a few different species of frogs described in Australia. Remember, three from 2022 were discovered in Australia by people using the app Frog ID. So you don't have to take photos of the frog. You can record their sounds and upload to the app and researchers can validate your like the identification for you. So yes, there are a lot of examples. New species of praying mantis was discovered in Victoria by a naturalist as well, people using a naturalist. So there, yeah, again, lots of new discoveries happening in the world, happening in Australia as well, happening in New South Wales. So as part of our establishing committee, our AXA New South Wales chapter, we organized a launch event. I believe some of you, some of the people here today might have joined us in June. Yeah, that was late June of this year. And we had amazing observations on that specific day. We also organized a BioBlitz. And then a BioBlitz, it is an opportunity to try to identify as many species as possible in a short period. So for this day, we had 2 hours, around 2 hours of BioBlitz. So we went around the area. So we was in Awabako Environmental Education Center. And we had lots of observations of invertebrates, lots of plants, lots of birds as well, fungi. But apart from the observations, I think it was also a great opportunity to connect with people who had similar interests. So we had people from land care joining us. We have people from bird life joining us. So bird watchers, environmental educators, people from the Awabako Center joining, people joining for the first time in a citizen science experience, and also people highly experienced in citizen science. So it was a great opportunity to connect, to talk about it, and to discover what other projects are available and going on around the world, around the year, along the year. So apart from those projects, there's a lot going on in Australia in terms of citizen science. Of course, if you check the Australian Citizen Science Association website, there is the tool Project Finder. So you can find there are over 600 projects in the platform. But specifically in New South Wales, there are seasonal projects, so they might change from year to year or time to time. But if you check the New South Wales website, Environment and Heritage website, you can find a list of citizen science projects and you can find which projects are available around your council. And you can find also different ways to participate. So iNaturalist is a great way, but if you're not a fan of technology, if you don't want to use apps or phones, there are other ways to get involved in citizen science. There is the Bat in Your Backyard, where the project leaders provide you with a kind of a sonar, a little device that you can plug on your phone. It's not an app, but like you plug on your phone. and it can record the sonar of bats, microbats flying around your backyard, so you can help with the surveillance of specific species. And if I live along the coast, there are projects to count turtles, specific species of turtles, and count koalas, and yeah, a lot of different projects and ways to get involved. So I highly recommend if you have a chance, if you're interested, if you sign up for the AXA newsletters, you might find out about some specific citizen science events that, again, they are seasonal and they might be coming up in the next few months. Uh, but apart from that, if you actively check with your local council, they might advertise citizen science projects or specific initiatives, local initiatives happening in that specific week. And then that might be an opportunity to get involved. I was in Sydney a few weeks ago running a moth night, and that was something that, we organized almost in the last minute, and we advertised with Sydney City Council, and then we had like a lot of people coming to the university for the first time. It was at the University of Sydney, so they came to the university for the first time because they saw the newsletter from the council. So that's my recommendation. If you have a chance to join the council's newsletter as well as the AXA newsletters, so you can hear from different citizen science projects and different ways to get involved and find out something that you are really interested in. You might love a naturalist, but you might find out that there you might love the koala count, for example. So there are different ways to get involved. And yeah, we'll have Thomas talking a little bit about a naturalist now.
Thanks, Larissa. That was fabulous. And I hope you all really enjoyed that quick squiz around citizen science projects in New South Wales and a bit of an introduction to citizen science itself. Really great. Thank you. Larissa, are you able to just pop into the chat some of the links and some of the things that you were just talking about so that everybody can have a look at those? And if you still have questions from the audience, then please do feel free to put them in the chat. If you think of more further questions for Larissa, you're still welcome to pop them in the chat now, and we'll come back to them at the end when Thomas has finished speaking, and we can all have a quick discussion about it. All right. So, talking of Thomas, I will now introduce Thomas Massalio, who is a botanist and ecologist based at the University of New South Wales, Sydney, and the National Herbarium of New South Wales. He recently finished his PhD on biodiversity informatics. Thomas, have you been conferred yet?
No, the gears of university administration grind quite slowly, so
Okay,
not
well,
quite yet.
you have my absolute respect for having finished, and I look forward to the time that we call you doctor once the university have conferred you. He's also previously worked for CSIRO's Atlas of Living Australia, which I'm sure he's about to tell us about as well. Thomas is heavily involved in citizen science. He's a site administrator for iNaturalist, which he's about to talk about, as well as, for Australia, iNaturalist Australia specifically, as well as a curator and forum moderator on the platform. He regularly organizes, leads, and participates in BioBlitzes and runs workshops and presentations teaching people how to use iNaturalist and get involved with citizen science. Please enjoy this presentation by soon-to-be Dr. Thomas Massalio.
Thanks a lot, Lynette, and thanks, Larissa. So pretty much the way I'll run this, like I do with a lot of these workshops, is I'm going to introduce you to iNaturalist, which is one of, if not the biggest citizen science platforms in the world, as well as in Australia. And so we'll basically go through a little bit of a tutorial for how you can get involved in iNaturalist, both with uploading your own records, your own biodiversity observations, but as well as using and browsing the data and the records that other users have uploaded. Feel free to please ask questions at any time in the chat, and I will answer those as they come in. So one key thing as well, just to note before I start, is that this workshop will be addressing iNaturalist in terms of the desktop version. So some of you who may already use iNaturalist may be familiar with it as a mobile phone app. The app is certainly great for accessibility and convenience when you're out and about. But I do highly recommend if you are at home, it's best to use iNaturalist in your browser on your computer, just because there is a lot more functionality and it's a lot more efficient use of your time to upload records through the browser. So you've gone out, you have taken photographs, you have taken sound recordings, whether these are in a remote tropical rainforest or the middle of nowhere in the desert or your very own backyard, and you want to keep a record of all of the species that you have seen. And for some things, you want to try and get them identified as well because you're unsure which species they are. Well, iNaturalist is a fantastic outlet for this, where you're able to engage with a huge number of other passionate citizen scientists and naturalists from not just around Australia, but from around the entire world. And many of these are not only amateur naturalists, but you also have taxonomists, museum and herbarium curators, literally world experts in their field that are able to interact with you and share their knowledge with you and identify records that you upload. So it's always quite an exciting platform to be a part of. If anything, because of the amazing people you get to interact with, a lot of like-minded people. So what I'll do first is I'll quickly just run through how to upload your records via the desktop. So we'll just go through the big green button that says upload in the top right-hand corner, and that'll take you to the upload screen. Now, as I alluded to earlier, it's generally a lot more efficient to upload your records via the desktop. So obviously, particularly if you're taking photographs with a digital camera, then you would download those photographs to your computer and upload them via this particular upload screen. But even if I'm taking photographs with my phone, I generally find that when I get home, I just download the photos off my phone onto my computer, and it's just a lot faster for me to upload them this way. Where if you're using the app, you're really limited to kind of uploading one observation at a time, and it can be quite a time-consuming process if you've been on a big walk and you've taken lots of photos that day. So if I'm on a particularly big adventure on a big trip somewhere, I can take anywhere between 1,000 and 2,500 photos in a day. Now, most of those are going to be on my DSLR, but a lot of those might be on my phone as well. So it's a lot easier to do it on the desktop because I can upload them in batches of 50 to 100 photos at once. So I will quickly show you how to upload both sounds and photographs. So what I have is I just have a Windows Explorer file open with the folder with my photos. So this is just a small batch of photos that I took earlier today. So I'm going to go ahead and click the first one and then hold down the Shift key on my keyboard and then click the last one. And so I've selected all of my photos and then I can just drag them across into the screen. And you're going to see they're going to start uploading. Now, while they're just loading in, my particular camera I use at the moment has quite large file sizes, so they take a little bit to load. I'm also going to show you what to do with audio files. So what I do is I use this particular online converter. So iNaturalist doesn't take videos, but it does take audio files. So if you encounter something like a bird or a cicada and you can't see it and you can't get photographs, but you want to take sound recordings, that's perfectly okay. Normally, what I will do is I'll actually take a video with my camera. I find that, at least for my particular camera, the audio quality from the microphone on the camera is actually far superior to the audio quality if I take a video with my phone. So what I do is I will take a video using my camera, and then when I get home, I will load it into this video to audio converter. So I grab the video here, I just again drag it into here. I'll set it to M4A, which is an audio file type. I'll select best as the quality, and you'll see that will start to load. I will paste that link in the chat for you. So as I said, that's the one I use. It's free to use, but there's a lot of the same kind of stuff out there. Now, while this one is loading, we will swap back and we'll go into our photos while that's loading. So you see here at the moment, I've got 18 photographs, but these do not correspond to 18 different observations, because the key thing when you're using iNaturalist is that if you take multiple photographs of the same organism at the same time, they all need to be combined into a single record. Whereas if you're then on the other side of that coin, if you're taking photos of different species on your walk, they need to be separate observations. So here, where I've taken multiple photographs of a particular plant or a particular organism, I need to combine those photographs into a single record. And this is very easy to do, and I'll show you how. So for something like this turpentine, for example, so here I've got 1, 2, 3 photographs of this turpentine. So that's Syncarpia glomulifera, relatively common tree species you can find along the east coast of New South Wales in wetter forest. And what I'm going to do is I'm going to click on that first one. You can see it's now outlined in green. I'm then going to hold down the Shift key on my keyboard. I'll then click the last one, and you can see it's now selected all 3 of those photos. They're all now outlined in green. And then I just click the blue combine button at the top of the screen. And you can see those photos have now been combined into a single record, and it says 1 out of 3. And then I can page through 2 out of 3, page through 3 out of 3. So really, really straightforward. We can go down and see the same thing here. I've got these 2 photos of the same plant. I can just click on the first one, hold down the Shift key, click on the second one, click combine, and they're put together. Alternatively, if you've just, this shift combine method is good if you've got a lot of photos. If you've only got a few photos of a plant, you can also just pick up the photos and drag and drop them onto the first one. So let's say for this particular one, this is a Raphiolepis. I want this to be the first photograph to appear in the record. All I have to do is go to the second one, pick it up, and dump it onto the first one. And then again, for our third photo here, pick it up with my left mouse and then dump it onto the first one. So now we have combined those 18 photographs into 13 records here. And so now we can go ahead and fill out some of the metadata. So you'll notice that for all of these records, 2 of the 3 fields have already been filled out. Now, 99.9% of the time, no matter what you're taking a photograph with, whether your phone or a camera, the date and time are probably going to be filled out automatically for you. If they aren't, for whatever reason, or if the date happens to be wrong, you can easily just click into the bar and you'll have a little pop-up that shows a calendar, and then you can just page through and select the date and time that you took the photograph. That one's simple. In terms of the location, you'll see this here. Now, again, in a lot of cases, this will be filled out for you automatically, especially if you're taking photographs with your phone. So if you do start to use iNaturalist and you're taking photographs with your phone, just make sure you check to have your location services turned on. If that's the case, then coordinates will be automatically embedded in the photograph for you. Some cameras will have automated geotagging where they will also automatically geotag the photos for you. But for many DSLR cameras, many digital cameras, they do not automatically geotag your photos. So you'll have to do that in one of 2 ways. One of them is that if the location bar is blank here, you can simply click here. You'll get prompted with a Google Maps window, which opens up. You can either search for locations in the search bar, just like you would do in a normal Google Map search, or you can just scroll in and out and you can physically navigate to the spot where you took the photograph. You can zoom in and you can click on the spot. You'll notice that when I click a spot on the map, not only do you get this central point, you get a red circle. This red circle indicates the coordinate uncertainty. So if you weren't totally sure exactly where you observed a particular plant or animal, you can just grab onto one of the handles on the side of that circle, and you can expand it until that circle covers all the possible areas where you could have observed that organism. You'll notice down here as well, so you've got the latitude and longitude are filled out. You can manually set this accuracy, so you see that will change the size of the circle. You can also then manually fill out location notes. So at first, when you click on the map, these location notes are just automatically populated by Google Maps, but I can go ahead and type in whatever I like there. And then once I'm done, I can click Update Observations. So you can also set the location notes or coordinates to many observations at the same time. So let's say, so when I click on this particular one, these were all observed in this one reserve this morning that I was at. And so I'm pretty happy with the locations for all of these. But what I'm going to do is I'm going to set the coordinate uncertainty and then the location notes for all of them at the same time. So all of them are in the same place. I want to apply the same notes and the same coordinate precision to all of them. And so what I'm going to do is I'm just going to click select all up here. So a little checkbox, I click select all, and you'll see that now all 13 records have been highlighted or outlined in green. And so then instead of clicking the location bar here on one of these individual records, I'm going to click it over here on the left-hand side where you'll notice it says edit multiple locations. So now when I click in here, any changes I make to the location data here will affect all of these observations. So I'm going to go ahead and type in 15 because that's generally the coordinate uncertainty that I apply to my records when I geotag them using a special app that I use. So this is the second way of geotagging things. I should briefly explain it. So the first one is I can go ahead and just navigate on the map here. But the second one is you can use a geotagging app on your phone. So on my phone, I have bought a geotagging app. Again, there are many, many different options that are available. The particular one I use is called Geotag Photos Pro 2. Costs about 20 bucks. And basically, when I start a walk, I click start on the app, and then it starts to track my location my entire walk. You can set it to continuous tracking, which is what I do, or you can set it to only tag your location every 5, 10, 50, 100 meters, whatever you'd like. Or you can also choose to tag your location every 5 seconds, 10 seconds, 1 minute, and so on. And once you have set that up, you just leave it running your whole walk. The only thing you have to make sure is that the time on your camera is synced to the time on your phone. You go ahead, you do your walk, you finish, and you click stop on the trail. You also then make sure you have the app downloaded on your computer. So once you buy it on your phone, you can then download it for free on your computer. And you just tell the app which walk you were on, and then you pick the photo folder, and it will then automatically geotag all of your photos for you. So I geotagged about 300 photos that I took earlier today, and it took about 45 seconds to a minute to automatically geotag them all, and it will embed all the coordinates. And the way it does that is it matches the timestamp on your photos to the timestamp on your phone when it was tracking you. Not only is it handy for geotagging your photos, but it also shows you a continuous path on your phone of where you're walking the entire time. So if you ever get lost, all you have to do is actually open the app on your phone and you can just retrace your steps and look at the exact path that you've been walking at whatever location you happen to be. It's something I've used multiple times when I've been walking in remote Australia, especially off track, and found myself turned around and not sure where the car was. All I had to do was open the phone and just retrace the path that it showed that I had already walked. So here I'm then going to also enter my locality notes. So here we're going to go Mashman's Quarry Reserve. I believe this suburb is St. Ives. So it's not necessary for you to fill out the location notes manually like this, but I always just like to fill them out with a bit more information because I generally find that the automatic locality notes that are generated by Google Maps are generally fairly coarse and pretty vague often, and I don't find them tremendously helpful. So I just like to fill in these manual location notes as well. So then I'll click the blue Update Observations button, and you can see now it has updated those notes in every single one of these observations. The last little thing I'll note there with the locations is that you'll have noticed there's another bar here, which is geoprivacy. So this is automatically set to open whenever you upload your records, but there's two other options there, obscured and private. So I'll jump to private first, and the reason for that is that I highly recommend to never use private where possible. This is a setting that you'd probably use in 0.01% of situations. A lot of people misinterpret what it means, so they'll often think that if they made a record on, say, private property, that they should select this option. This isn't really the case. It has a fairly idiosyncratic meaning within the context of iNaturalist. If I were to select private for this particular record, what happens is that all location information is completely destroyed from public view. So anyone else viewing this record, not only can they no longer see the coordinates of the record, they can't see the state, they can't see the country, they cannot even see the continent that the record was made. Now, this has a few repercussions. It means that it becomes a lot harder for people to find your records. So many people that are looking to identify records for other people are explicitly filtering down to a particular place. So generally, when I identify records for other people, I first filter down to records just from Australia. So if you had observed something in Australia, but you set the geoprivacy to private, that record would not show up in a search for records from Australia. And the second consequence of that, well, so one consequence of that is that it means your records become a lot more difficult to find. What that also then means is that they're less likely to be identified, and it means that your records won't go into the Atlas of Living Australia. So for those of you who aren't familiar with it, the Atlas of Living Australia is Australia's national biodiversity database. It collects records, so it's a data aggregator. It doesn't generate records itself, but the ALA is an aggregator that collects records from many, many different sources. So this includes iNaturalist. It includes other citizen science platforms like Frog ID that Larissa mentioned earlier. And it also includes all records from Australia's museums, from our herbaria, from professional surveys done by state and government, federal government employees. So it is the main hub of biodiversity data in Australia. But if you set your records to private, it means your records will not go into the ALA. So really, it gets used in very exceedingly rare circumstances. So for example, if there was, I'll use a North American example. So there are a few exceptionally rare and heavily poached groups of organisms in the Americas, a few groups like particular reptiles, a few particular groups of cacti that are exceptionally heavily poached. And in those particular cases, obscured probably isn't quite good enough. And I'll explain what obscured is in a second. And so for some of those, they get set to private as that additional level of security. But what you can do, if you like, is to set your records to obscured. So obscured is a nice middle ground. So what happens if you set one of your records to obscured? Well, you will have entered the true coordinates, but anyone else that is publicly viewing your record, what they'll see is a big box around your true coordinates. The box is 0.2 degrees by 0.2 degrees, latitude by longitude. That's roughly equivalent to about 500 square kilometers at the equator. It gets bigger, a little bit bigger and a little bit smaller as you move towards the poles, but it's generally a pretty big area. That box has a diagonal of somewhere between 25 and 32 kilometers. And then there'll be a randomly generated point inside that box. So for anyone else that's viewing your record, they will just see a randomly generated point and they'll know that the true coordinates are just somewhere inside that big box. Also, the date and the time get stripped off the record. So all they'll see is the month and the year. And any specific locality notes that you've written, like I have here, will also be stripped from public view. And anyone that then also identifies your record, the date and timestamp will be stripped off those as well. So people use this for a variety of reasons. One of the main ones that people will manually obscure their records is for privacy reasons. So for example, if you're making records on your property from your backyard or from your workplace, and you don't want people to know where you live or you work, then you can go ahead and set this to obscured. Also, let's say you observe a rare orchid, and maybe the orchid isn't deemed to be sensitive by the state government, but you're a little bit worried that it might be poached or collected by people that may be looking at your record. Again, you can go ahead and set it to obscured, and it's a nice extra level of security in terms of the location data. Now, when your obscured records go into the ALA, the only downside is that if you manually obscure one of your records, it'll still go into the Atlas of Living Australia, but the ALA will never give out the true coordinates of your records to any researchers or conservationists or other scientists that are trying to access them. So that's just one thing to keep in mind. If you do want your records to be accessible by researchers and by scientists, then manually obscuring them does provide a little bit of a barrier to doing that. There are other ways that those researchers can get access to those records directly through iNaturalist by directly contacting you, but it is an option there. One other thing is that many species on iNaturalist will actually have their records automatically obscured and you have no choice in the matter. So each of the state governments will provide a list of all of the species that they deem to be location sensitive. And the lists are quite different across the different states and territories, ranging from Tasmania, which just deems 4 species in the entire state to have sensitive locations, all the way up to Western Australia, which has thousands of location sensitive species, and then everyone else is somewhere in between. And then when I was working at the ALA, part of my job was to implement those statuses on iNaturalist. And so for all of those species, whenever you upload a record of them onto iNaturalist, they just get automatically obscured and you don't have a choice. So just one example of that is gang cockatoos. So if you observe a gang cockatoo anywhere in Australia and you upload a record to iNaturalist, the location will be automatically obscured. Now, when those records go to the Atlas of Living Australia, the ALA staff and the data managers, they are able to give out the true coordinates of those to researchers and scientists as long as they have a valid use case for them. So the last bar that we have to fill out here, the last piece of metadata, is the identification for each of these records. And you can do this in a couple of different ways. So one of them is, well, if you know what species you're dealing with, you can just go ahead and type in the name, whether that's the scientific name or the common name. So for this particular one here, this plant, this is elderberry panax. It's Polycia sambucifolia, pretty common species in southeastern Australia. I know what it is, so I can just go ahead and start typing the name, and you'll see a little drop-down box will appear. You just have to make sure you click into that drop-down. Don't just type the name. And so I've gone ahead and I've identified my record. Totally fine. Now we can go ahead to this second one. Let's say that I didn't know which species this was. All I knew was that it is a plant. That's totally fine. I can go ahead and type in plants as my ID, select that from the dropdown, and away I go. Now, it's really important that even if you have no clue what something is, please fill out even a course ID. It sounds a little bit stupid, but like let's say you see some kind of worm-like animal and you have no clue what it is. It could be an insect, it could be a flatworm. You've got really no clue. You can literally just type in animals as your ID, and that is totally fine. And that is better than leaving it blank. Why is that? Because just like many people will explicitly search for records by filtering down to a particular place, many people will explicitly search for records by filtering to a particular taxonomic group. And so if you leave the identification bar blank, it makes it a lot harder for those people that are filtering records to find your record. Whereas if you type in even a course ID, that's totally fine because here I've identified it as a plant. Well, there are heaps of people who are filtering records to plants, and so it just lets them see your record. What I can also do as a third option, if I have no idea, is I can see what the AI suggestions are. So I'm sure that at least some of you will probably have used something like Google Lens before or a similar machine learning piece of software. Basically, iNaturalist has a software called computer vision, which is an AI program where you can click into the species bar and it will give you a guess of what it thinks the species is. Now, just like any similar kind of AI tool, it's really important to take the suggestions with a bit of a grain of salt and to assess how legitimate they seem yourself, rather than kind of just blindly accepting one of them and assuming that it's correct. For some groups of organisms, the AI suggestions are fantastic. If you take a photograph of pretty much any bird from anywhere in Australia, even a rubbish photograph from far away that isn't in focus, it will get it for you. It's exceptionally good. And the reason for that is that for groups like birds, the AI model has a huge amount of training data. And so whenever you upload photographs onto iNaturalist, those also all contribute to training the AI model within iNaturalist that is then able to suggest species suggestions. So for things like, say, a magpie, there are literally tens of thousands of records of magpies on iNaturalist, hundreds of thousands of images of them. And so the AI is exceptional at recognizing photos of magpies. Makes sense, right? Whereas once you start to get more obscure groups, like a lot of, say, rare groups of plants, obscure invertebrates, or particularly organisms from more remote parts of Australia where there aren't a lot of records, places in, say, Queensland, Western Australia, Northern Territory, well, then the suggestions aren't going to be as good and often they'll be wrong. And in particular, if I take photographs of, say, a pretty rare organism and it doesn't have many records on iNaturalist, it is literally impossible for the AI to suggest the correct species. Because until a particular species has roughly 100 photographs, it cannot enter the training data for the AI model. So if I'm observing a particular species that's quite rare and just has a small handful of records on iNaturalist, it is impossible for the AI to correctly suggest it, but it will still suggest other things that look similar, but that are nonetheless wrong. So it's totally okay to look at the AI suggestions, and they can be a really good starting point for you. But just have a bit of a think about whether they seem legitimate. So I'm going to go ahead and see what the AI says for each of these things. So I told you that this particular species here is Syncarpia glomulifera turpentine. Let's see what happens when I click into the bar. So it says loading suggestions, and then here we go. It is spot on. It's correct. So this is a pretty common plant. There are hundreds, if not thousands, of records of this plant species on iNaturalist. It has a lot of photos. It can recognize this species well. So first up, you'll notice it says we're pretty sure it's in this genus, the Syncarpia. That's correct. And then it'll have top species suggestions, and that one is correct. So I can go ahead and click on that. Totally fine. Same thing here. We can see this species. For anyone who does bush care, you probably recognize this species. It's Ochna cerulata. Gets called Mickey Mouse plant. It's a very, very nasty weed in Australia. I'll click in the species name bar, and there we go. Top suggestion is Ochna. Now, what you'll notice will happen is that the fewer suggestions it offers, the more confident it is of those suggestions. That doesn't mean that they're correct, but it means that the AI model is more confident. Whereas if there's something where you have a lot of suggestions, up to say 7, 8, or 9 suggestions, that means it is less confident. So generally, as a good rule of thumb, I like to do is if I'm not sure whether an AI suggestion is correct and there are very few suggestions, you can probably safely pick it. If it's wrong, it'll be corrected by someone else. But if it has a lot of suggestions, I would not pick one of them. So here, for example, you'll notice for this particular plant, well, it's got way more suggestions here. And you'll notice that for the section where it says we're pretty sure, it's actually not even sure of a genus. It's pushed it all the way back to tribe. So this is one where I definitely wouldn't take one of these automated suggestions if I didn't know what the plant was already, because it's clearly not very sure. Now, as it happens to be for this one, the top suggestion is actually correct in this particular case, but you'll notice it has far lower confidence than for the other records. So I can just go ahead and type in that one, and that is correct. So we can go ahead and fill these all out. These are all correct. This one I know is Brainiea oblongifolia, and again, it gets it correct. Now, these ones are interesting. So these ones are actually leaves of a plant species, Pittosporum undulatum, but you'll notice that it doesn't actually offer a suggestion of the plant for them, because what I was actually taking a photo of is these galls on the leaves. So these galls are formed by a leaf mining fly, Phytolyria myza pittosporophyli. And there are so many records of this particular galling species now that actually when you take photographs of the leaves, more often than not, it will suggest the gall because it assumes that if you're taking a close-up photo of the leaf, that's what you're interested in. So for this one, you'll actually see so here, you'll notice the galls are very prominent. And so the AI can easily tell what you're focusing on. For this one, you'll see there's only a single little gall here on this photo. It's a little bit, there's a little bit of glare. There's some other feeding damage from other insects. So you can see it actually has a second suggestion here. As it is, that plant suggestion is not correct, but it still does suggest the gall as the top option. Now you can see here again with a bit more of the leaves in view, it actually does suggest the plant as well as a suggestion. But I'm just going to focus on the galls for all of these. So you can see for particular records, it will only suggest the gall. For others, it will also suggest, okay, so here it will suggest the plant. And generally, what you'll find is if the photo pretty much only has leaves which have galls on them, it'll just suggest the galls. But you'll notice for this particular one, there are actually some leaves here that don't have the galls on them, and then it'll suggest the plant as well. So I can just go ahead and select all of these and then go down to my last one and see this is another one here. So again, the top option is actually correct here, but you'll notice it's far less confident of that fact because it has offered up so many suggestions here. And it makes sense that it's less confident, right? There's no fertile material here. These are relatively generic looking leaves, so there's a lot of different species that it could possibly be. So, but I'm going to go ahead and select that top option because that is indeed correct. Um, and I'll just very briefly answer some questions here. Yes, so sometimes, yeah, sometimes I have entered IDs as just life as well. And again, it sounds kind of weird, but you can put in an ID of life. So I can show you what that looks like. So I can actually just enter an ID of life. And again, that's still better than just leaving it as a blank unknown because generally when things are left as blank unknown, they kind of tend to slip between the cracks a little bit. And Lynette also said, I've heard some people say if they put in an incorrect ID, they feel like they're more likely to be corrected. I feel like that is correct, but I wouldn't necessarily recommend it as a strategy because you do end up with some particular groups of organisms where they tend to get ID'd a bit less, and so it may end up sitting there for a while, being explicitly incorrect. So, if there's something like a bird or a common plant, your ID is likely to be corrected very, very quickly. But if it's a bit more of, say, an obscure insect or something like that, it could potentially sit there for months or even years waiting for an expert to look at it. And so in those cases, an intentionally incorrect ID, as tempting as it may be, it may end up sitting there for a while. But I do broadly agree with you that if someone puts a kind of egregiously incorrect ID, people tend to be a far more quick to jump on it and correct it. So that pretty much covers everything in the upload screen. Yeah, Larissa?
Thomas, can I ask a quick question about obscuring observations? I have a friend from South Australia, and he has a really big backyard, and it is on the bottom of a national park, like on the border of a national park. And he uploads observations from his backyard like every week, has like thousands and thousands of observations. But because they are from his backyard, he obscured all of them. And that's a way to protect his privacy, like let's say if somebody wants to come and have a look at the flowers or like the pollinators or things that like are usually active in the national park, but they are coming to his backyard. So yeah, what do you think of like obscuring in that situation, like for privacy?
Yeah, I think it really just varies from person to person. So I upload a lot of records from my own backyard as well.
Mm,
I never obscure any of them. But I guess, I'm obviously in a privileged position where I don't, there are a lot of people that I know. There are several people I know in iNaturalist who unfortunately have had situations with, say, a stalker or other similar personal situations where their privacy was really important. And so in those cases, of course, some obscuring observation was a really important thing for them to be able to still engage with iNaturalist and to feel safe. Um, for me personally, I'm obviously lucky enough to not be in a situation like that, and so I have never really felt the need to personally obscure my own records. But yeah, if it's something that you don't feel comfortable with, having people know exactly where you live and that particular species occur on your property, then yeah, definitely you can go ahead and obscure them. It just is that downside, as I mentioned, that when they go into the ALA won't be able to give out the true coordinates of those to any legitimate researchers or scientists. Um, so yeah, I guess it all just comes down to that individual need and those individual circumstances. But certainly, I highly recommend if it's something that you feel that you personally need, then definitely utilize that feature.
yeah, awesome. Thank you.
So the final thing I'll just do before I hit upload on these is we'll go back to our sound file. So that would have loaded a long time ago. Once that has finished converting, once it's finished loading, I just click the blue convert button and then I click download, and that will download the audio file to my computer. So that's now in my downloads. So I can actually go back to here and I will drag and drop that audio file into the upload screen. The only downside here is you'll notice that all of the metadata doesn't get filled out, so you'll have to do that manually. So I'm going to go ahead and just very quickly check my phone where I took this video. So I took this at 9:23. So I can go ahead and copy that and then edit that. And I know that I took this um, in the reserve, and here's a one that I also did at 9:23. So I can go ahead and pop in the location to be the same as one of these. This one was just there. So we'll go ahead and put that one there. We'll set that to 15. We'll go back into this one, copy those notes. I'll add it to a couple of projects. And this one was a cicada. So relatively straightforward to do those audio files as well. So now I've got all of my records. I'm ready to go. I just click the green submit button at the top right-hand corner, and away those observations go. And they're all uploading. So it's as simple as that. And you'll notice that, as I said, I did many photos at once, and that was only a small batch. You know, if I'm on a big trip, I will regularly do those in batches of anywhere between 50 and 100. And it's a very efficient process. So now those have all been uploaded. I can go ahead and wait for people to either ID them or to confirm my IDs. And in the last, let's say, 10 minutes before we open it up to some questions, last 5 or 10 minutes, I'll just show you how to quickly navigate other people's records. So even if you upload very few records yourself, or if you never upload records yourself, that's totally fine. There is a huge wealth of existing data out there that you can have a look at. So every single day across Australia, 10,000 new records get uploaded to iNaturalist day after day. Across the world, it's about 150,000 to 200,000 new records get added every single day, day after day. So we can go ahead and we can click this Explore button at the top. And so this will show me at the moment all 287 million records from across the world. Let's narrow that down to, let's say, New South Wales. And we can see for New South Wales, there are about almost 3.2 million records. Let's say narrow that down to butterflies. I can see the 58,000 records now of butterflies that have been uploaded to New South Wales. So you can slice up the data in many, many different ways. There are hundreds of different ways that you can filter the data. This includes both with these filter boxes here, which give you some very basic options, but there are also many ways which, if any of you want to find out, feel free to email me or come to one of my advanced workshops. You can also manually edit the URL up here in hundreds of different ways to do a lot of really complex filtering of the data, but we can stick to some basic ones here. So let's say, for example, I'm interested in butterflies in New South Wales in the summer months. So I can actually filter this just to butterflies that have been seen in December, January, and February. So I can go ahead and I can say, okay, so far for New South Wales and iNaturalist, 20,000 records of 181 different butterfly species have been observed thus far. I can swap to a map view, and so I can see exactly where they are. Look, no surprises, Western New South Wales doesn't have many, but a lot of the records are concentrated around population centres like Sydney. So I can go ahead and I can see, okay, there's quite a lot of records here. I can then say, click this bounding box and I can draw a box around this kind of Greater Sydney area and filter it down even more. So now it's a bit over 4,000 records across 90 species. I can click on any of these records at any time to go to that individual record and find out more details. I can sort back to the grid view, and instead of looking at the individual observation view, I can look at the species view. So for the observations, they are ordered by default from the most recently uploaded to the oldest. So as I scroll down, these are all records that would have been uploaded from today. I can swap to the species tab, and this will show me records ordered from the most commonly observed butterflies to the least observed. So this isn't necessarily the most common butterflies, keeping in mind it's the most commonly photographed and uploaded to iNaturalist. So you can see up here, the most observed butterfly from the summer months from that bounding box around Greater Sydney is the orchard swallowtail. No real surprises. This is a very large and conspicuous butterfly. It tends to be relatively slow flying, so it tends to be one of the easier ones to photograph. And then we can scroll down. You can see other big, charismatic butterflies like monarchs, swordgrass browns, painted ladies. And we can kind of scroll all the way to the bottom and start to see the more rarely observed ones. So things like a lot of these, you'll notice a lot of blues. So these tend to be very small, very fast flying butterflies that are difficult to photograph. So these are the ones that only have a single observation thus far from Greater Sydney in the summer months. You can also go to the identifiers tab and see who has been identifying these butterflies to try and figure out who you can contact, for example, if you need to get an identification on one of your records. And you can also look at the observers. So they are ordered by default by observations, but you can also swap over to the species tab. And so you can see who are the top observers of butterflies in Greater Sydney in those summer months. As I said, you can slice up the data any way that you possibly like. So we'll do just another little test to just show you the power of this. So let's say that you're a seed collector and you're interested in going out in collecting seed for peas. So we're going to just filter to New South Wales again. And now we're going to filter to Fabaceae. And I want to go out and I want to collect seeds for some of these peas for the native nursery that I run, but I'm not interested in acacia. So I'm going to swap to this species tab. I'm going to find an acacia, and you'll notice in the top left-hand corner here, there's these three little lines. I'm going to click on that button, and I can actually go ahead and click the minus here, and that excludes acacia from my search. So now I'm searching for all peas in the family Fabaceae in New South Wales, but I've excluded acacia. Then I'm going to go to the filters. I'm going to say, okay, I am only free to do seed collecting in August. So I'm going to swap my months observed to August. So now I can see so far there's about 11,500 records of peas across 300 species from New South Wales that have been uploaded in August. But I then want to further filter to only the ones that have been annotated as fruiting. So the way I can do that is by manually editing the URL. So I'm just going to do this very quickly and not show you how to do it because this is a bit more of an advanced thing, but I just want to show you that the capability exists. And so I'm going to manually edit the URL here. And so I'm going to filter down further. And so this is now showing me just 113 records from New South Wales across 44 species from the month of August of pea plants, not including acacia, that have explicitly been annotated as having fruit. So you can go ahead and you can see, for example, here's a Dilwinia, and you've got fruits on those Dilwinia. Here's an Almalea, which is a much rarer genus of pea, and you can see the capsules there. You can see this Erythrina has a pod with fruit. Here are fruits of a Crotalaria that actually I observed. Again, here's pods of this Swainsona, Pultonea. You can see pods in the background here of this Gomphilobium. There are pods in this Hovia. So this is just a really nice way. So it's a really nice tool. And again, you don't ever have to upload any records yourself, but there are so many thousands of millions of records that it's a really nice way for you to go on and find records and inform your own searching and your own adventures. So another way you can do this is looking at the profile page for each species. So let's say that I'm really interested in waratahs and I really want to see a waratah in flower. What I can do is I can type in waratah here, then I'll click the About page, and I'll get taken to the profile page for waratahs. And then you'll notice there are these really handy charts. So I can say, okay, most people tend to observe waratahs between August and November. Look, there's records of waratahs all around, but most of them tend to be August and November. And then if I click this Flowers and Fruit tab, you can see this is where people have gone ahead and annotated records. And you can see the red curve here is flowers, and you can see it peaks in September or October. So now I have this really useful information that if I want to go out and I want to photograph a waratah in flower, I should probably head out in the months of September or October because that is the greatest probability that I'll find a flowering waratah, because that tends to be when most people are photographing and annotating flowering waratahs. So it's just a really nice tool that, as I said, you can go ahead and filter the data in a huge number of ways, and it allows you to go ahead and use data used by people from all around the world. You can collaborate and communicate with these people as well. So you can send personal messages, you can leave comments on their records. There are many friends that I've made through iNaturalist. There are many collaborators and colleagues who I have published research with, who I have met through iNaturalist. So all around, it's just a really nice tool that not only can it inform and be useful for you at a personal level, but as I've mentioned as well, your records will feed into the Atlas of Living Australia. And from there, they get used in hundreds, even thousands of different research papers from all around the world. So it's a really nice way for your records to have a real world impact as well. So we're getting towards the end of the presentation. I could keep talking here and showing you all kinds of interesting functionality for quite a long time. But for this last 15 minutes or so, we might open it up if people have any pressing questions, whether they're relevant to iNaturalist or to citizen science more broadly.
Fabulous. Thank you, Thomas. Oh, and just as I was about to invite further questions, perhaps before we go to those questions, I'll just say that you're welcome now to turn on your camera, maybe even your microphones. And that we are recording this session. So if you are not keen to be identified, by all means, keep your camera off. And you can even change your name to something anonymous as well. If you have questions for either Thomas now or Larissa, or more broadly for citizen science, we've got other committee members in the audience, including our esteemed national chair, Stephanie von Gavel, is here as well. So welcome, Stephanie. Thanks for your message that I saw pop up as well. So do feel free to pop them in the chat. Hey, there she is. Thomas, would you like to answer Simon's? I haven't read it yet. I presume it's for you. But
Yeah, absolutely. So Simon said, do you have any tips for capturing species characteristics that might be important to ecologists? Things like root, fruit, size, habit. Should you try to capture multiple data attributes? Absolutely. If you have the time and the interest, then the more data, the better. Especially because you don't know what data might be useful 5, 10, 15 years down the track, right? And this is true of both citizen science records, but also natural history collections records in herbaria and museums. There are all kinds of amazing ways that scientists are now using data that was collected sometimes decades or even centuries ago. So absolutely, if you want to go ahead and take measurements, take photos of as many features as you possibly can, extra ecological information like pollinator relationships and things like that, it is all going to be useful to someone. It might not be useful immediately, but absolutely it will be useful somewhere down the track to someone in some way that we may not even be envisioning right now. So certainly as much information as you possibly can cram in. You can leave that information, well, it can be in the form of extra photographs that you append to your records. You can leave comments on your records, adding additional context and information, or you can fill out these extra data fields, which include annotations and observation fields. So absolutely try and pack as much information as you can into all of your records.
thanks for that, Thomas. I have a question, Thomas. What about if the opposite is true? Like, for example, say, so my specialty is frogs, and if I were to see a frog that wasn't known from an area before, but I definitely knew that it was there, and even if I was just an enthusiast rather than anything else, what do you think about records that don't have much information to offer at all?
Yeah, definitely. So look, any data is better than no data, I think is the basic way to put it. And even if you're recording really common species, it's really important because if people didn't record common species, they wouldn't appear as common in the databases. And so, you can sometimes have a bit of the bystander effect almost, where say I go out and I see common birds on my walk and I think, well, lots of other people will have already photographed those common birds, so I don't really need to do it either. But then if everyone thinks like that, then no one will be recording them, and you don't get an accurate reflection of the abundance of these different species. So every record has value as an occurrence because it tells us that this species occurred at this place at this time. And all of those records can feed into things like spatial distribution models. They feed into understanding how populations might be changing over time. There are plenty of studies and research that have used thousands, tens of thousands of photographs of even common species to understand things like different color morphs or how phenology might be changing over time. So that's another really key thing. As we progress over the next few decades and we're going to be hit by climate change, you'll have, say, plants flowering at different times of the year. And so if people aren't recording even common plants and showing when they're flowering, when we're fruiting, we won't have an understanding of how that phenology is actually shifting over time. So, absolutely, even if you know what something is, and even if you think it's a common species, it definitely has value.
Yeah, thanks. I think it's worth mentioning as well that, when scientists are designing studies that use citizen science data, they do take into account things like what chances are that people spot them, whether things are more obvious. They call it detectability, right? The idea that some things are more obvious than others. So yeah, I agree, Thomas. I think bang it in there, whether you're sure or not. Another question from Simon: Do you carry or use a black and white scale reference like archaeologists use?
Yes, I do indeed. And if I briefly share my screen again, so you'll be able to see a photo that I took today. So I use a little card, a little measuring card that has color scale measurements. Where will it be? Here we go. So so I was out in National Park earlier today collecting plants. This is a particular weed that I know the genus already, and I knew beforehand that for this genus, it's really important to get floral measurements, particularly of the ratio of the bracts to the floral tube, and also measuring the size of the floral lobes. And so this is just a little card that I carry around. You can see it's got measurements here. It's got color reference. And so this was a little card that I got from the Wild Orchid Watch, which is a citizen science project that runs out of South Australia. You can pretty easily print off your own ones. Ferox australis, I'm pretty sure Larissa has them as well. So it should be pretty easy to find ways and designs online that you can either print them yourself or get them printed from a provider. Um, they're just cardboard, so it's super lightweight. I just have them in either my pocket or in my camera case, and they're really handy to carry around, particularly for groups like if you're photographing plants, things like grasses, for example, measurements are really, really important for ID. So they can be a really handy thing to carry around.
Yeah, thanks. And Stephanie's put a link to one in the chat as well there. So there's plenty of options for you to get around it. Fantastic. Well, if anybody's got any further questions, shout now. You've got about 10 seconds left to raise your hand or type it in the chat before we finish up. Larissa, there's always one. Go on, Larissa.
It's not a question. Yeah, it's not a question, but I just wanted to say that I love the Explore tool from iNaturalist. So every time I go on a bushwalk, I just moved to Melbourne. So every weekend I go on a bushwalk on a national park here. I always use the Explore tool to see what people observed around the area. And I always try to find echidnas, and they're very hard to find. So I always try to see where people uploaded, the observations. And that's something that was very useful. 2 years ago, I went to Tazi, and I am from Brazil. So that was my dream. Like I wanted to see a platypus, like in the wild. And I used the Explorer tool to find like where people were uploading observations of platypus in Hobart. And all the observations were coming from the same river. It was a creek. So all observations were around the creek. And I was like, okay, I have to go to this place. So it's going to be easy because all of them are on the same place. So I drove there and I saw three platypuses on the same day. So yeah, that's something that I love about iNaturalist. As Thomas was saying, like if you're not into taking photos or if you don't want to upload anything, you can still take a lot out of the app, like you can discover so many things from using the app.
Yeah.
Um, yeah, so I think Stephanie's saying she had a similar experience trying the ghost fungi, the ghost
Glow in
mushrooms.
glow in the dark fungus. Yeah, great one. Yeah.
Yeah.
Fabulous. All right. Thanks, Larissa. And thanks, Jim and Carlos58 for your kind words in the chat as well. We've really enjoyed having you. Larissa, can I get you to just quickly pop in the chat there some links to the Citizen Science newsletter, the AXA newsletter. And if you want to follow AXA on social media, we've got Facebook, LinkedIn, and Instagram pages. So do pop us a follow and stay abreast of things that are happening for citizen science in New South Wales. All that's left is for me to say thank you very much to Larissa and Thomas for your fabulous presentations tonight. I'm glad to hear that you all got something out of it, and do feel free to come to our next events. I don't think we've got anything in the pipeline, have we, Larissa, for just yet, except for Christmas and resting, maybe? But
Yeah, Christmas videos, if you want to observe
yeah.
some, upload to our naturalist. Yeah, we want to organize an AXA New South Wales event for next year, like an in-person event, like another BioBlitz. We will keep everybody posted. If you join our newsletters, you get you get the dates when we have everything organized.
Yeah, fabulous. And there's lots of resources on the Australian Citizen Science Association website as well. So have a look at citizenscience.org.au and you can find some more things about citizen science more broadly on there, as well as more local stuff as well. So thank you all so much for joining us this evening. We've had a wonderful time showing you around some of what we know about citizen science, and I hope we'll get to see you all again soon. And thank you very much for your fabulous questions and your great discussion, and I hope that it's been useful to you. All my best, and catch you again soon, I hope.
Thanks, everyone.